All right, good morning, everybody. Welcome to our last lecture. You guys have made it. You made it to the end of the semester. It's amazing. I hope that biochemistry really helps you in your future classes. We're going to finish off with one other metabolic situation, this idea of ketogenesis, talking a little bit about these ketone bodies. I did post a couple of resources, so we're going to learn about ketone bodies in terms of these molecules that can get used for energy. Okay, so they are a metabolite that can be catabolized and broken down to generate ATP. We'll talk a little bit about metabolic conditions when you have too many of them made, but they're also being used in terms of ketogenic diets to help supply energy for different cells, because you have to start thinking about the variety of cells that are in your body. They all have different energy requirements, and they also have different abilities to break down certain molecules versus the other. Okay, so in terms of the brain, we've talked a lot about glucose homeostasis, because your brain's dependent on glucose for energy. We're going to see that ketone bodies can also get used by the brain because they can't use fats. Okay, so it has been used in treatment options for various neurological conditions. So just something to start thinking about in terms of taking different approaches when we're considering what's going on in the body. All right, so we're going to talk a little bit about the biochemical pathways, talking about how they're made, talk about how they can be used, and we'll see when they're actually made in diabetes as well. Who hasn't had breakfast yet this morning? Does anybody know? Okay, you're all undergoing ketogenesis right now. There are fats being broken down in your body. Those ketone bodies are going to get supplied to your brain for energy. On Tuesday, we talked a little bit about gluconeogenesis, so your liver's probably also breaking down all that glycogen, synthesizing glucose in gluconeogenesis. All of these things are going to help provide that energy for your brain to hopefully pay attention during today's lecture. Okay, so definitely think about how can you fuel your body in order to provide it the energy that it needs to carry out all those important functions. And then we're going to end today's lecture just talking a little bit about, we'll come back to some blood and urine tests, talking a bit about how you can use the levels of different molecules to assess whether or not there might be some sort of underlying metabolic condition. And really, you know, this is going to help sort of integrate all of these pathways, and I'll do my best to try to review stuff for the final exam, which is coming up. So once again, this skeletal diagram is going to be provided on the final exam, so you can go through, you can add in the hormones and which pathways they're going to turn on and off, the signaling pathways. You can talk about which enzymes are going to be phosphorylated or dephosphorylated. Maybe you want to annotate in what molecules can stimulate a pathway versus inhibit a pathway. So maybe if you have a green pen and a red pen, you might want to do green for go, red for inhibit. Whatever you need to kind of help do that knowledge dump before you actually look at the questions. Okay, and then you can cover up your answers for multiple choice questions, come back to this, think about everything you know about the various pathways, and then look at the answers so that you don't get confused by some of the distractors. So what we're going to talk a bit about today is this final pathway in here, where we start with acetyl-CoA, and we're going to be synthesizing these ketone bodies. We've already seen how it can be used to make cholesterol and those fatty acids, and really these ketone bodies come from acetyl-CoA, which comes from all of our fuel molecules, right? Pyruvate, which came from carbohydrates, can produce acetyl-CoA. Amino acids, some of them can make acetyl-CoA as well, or the glucogenic ones that we talked a little bit about briefly on Tuesday. And then when your fats get broken down, they also supply a huge amount of acetyl-CoA. So remember when we were doing those ATP calculations, where we started with a 16 zero chain, we were getting 8 acetyl-CoAs made. So lots of acetyl-CoA that's going to get made from our various molecules. So really when ketogenesis is going to kick in is when your fats are going to be broken down, as well as maybe if you're in a ketogenic diet and you're consuming a lot of protein as well. Okay, so it's really going to happen. Sorry, hang on one second. Let me just turn this off. One sec, I'll just mute that. All right, so we're talking about when we have a ton of acetyl-CoA that's going to get made, so either from your fat reserves getting broken down in beta oxidation. We also have some amino acids that can also be broken down to produce acetyl-CoA, as well as glucose. Glucose also produces acetyl-CoA, but really when ketogenesis occurs is when we're actually in the absence of glucose. Sorry. So when our fats are being broken down and our amino acids are going to be broken down, remember this is when we're in the catabolic state, so we have our hormones like glucagon and epinephrine. Those are going to be those catabolic hormones that are going to be signaling through our GPCR receptors, and it's going to produce a huge amount of acetyl-CoA. So really, when we're talking about ketogenesis, it does arise due to this large amount of acetyl-CoA. So it's when the amount of acetyl-CoA rises, and we can't process it or use it to generate a lot of ATP in the liver. Okay, and we're going to see what molecule it's going to be dependent on for that to happen. Then these ketone bodies can actually leave the liver, and they're going to get used as an alternate fuel source. Okay, and so we'll see how that happens and what they get converted back to. Okay. So we've got amino acids that can also get broken down. Again, you don't need to memorize which ones are glucogenic amino acids versus ketogenic amino acids, but there are some of the more hydrophobic ones. When they get catabolized and when we're removing that amino group, usually they come in and produce acetyl-CoA, and they can also contribute to the production of these ketone bodies. So we're going to remove that alpha amino group that we learned about at the beginning of the class, and then the carbons on those molecules will enter into the metabolic pathways that we've looked at. The urea cycle is going to help with the excretion of ammonia, and then the production of those acetyl-CoA molecules could help with energy production to make ATP, or we could either generate glucose if we're talking about gluconeogenesis in the liver versus ketone bodies, which are also going to get made in the liver. Okay, and so we've seen lots of examples of amino acid metabolism, right? That modification of the structure can help them actually come into these pathways and get used for energy or to make other metabolites that the cell's going to need. Okay, so again, you don't need to know that, but just appreciate that certain amino acids come into the pathways. Last day we looked at sort of the structures and how they come in to help produce the various molecules. Really, if you're starting sort of halfway through the citric acid cycle, we're going to go around to oxaloacetate, which can be used to make glucose in gluconeogenesis, versus the ones that will enter in sort of a little bit earlier on at acetyl-CoA. All right, so we've got excess amino acids that are going to get degraded in the liver. We've got the carbon skeletons that will get used for energy, maybe entering as pyruvate or acetyl-CoA or other intermediates. And depending on whether you're talking about an essential amino acid, so if you're on maybe a high protein diet or you're taking protein supplements, you'll see that some are essential amino acids, meaning you have to get them from your diet, whereas those non-essential amino acids, those were the ones where we're taking those metabolites within our pathways and then converting them into the various amino acids. Okay? We've got some that can be used to make glucose, and then others that can be used to make our ketone bodies. All right, so let's just do a little Mentimeter question in terms of if we actually do want to get energy out of that acetyl-CoA that might be made from glucose or fatty acid breakdown or our protein degradation, what molecule are we going to need in order to generate all that ATP from acetyl-CoA? All right, so we've got most people on the right track. So oxaloacetate is going to be that sort of limiting factor that's going to help us process all of the acetyl-CoA that might be coming from the various sources. Okay, so if we're thinking about our pathway, so here's our acetyl-CoA. Remember, when it comes into the citric acid cycle, we're going to be joining it with oxaloacetate. Right, those two molecules are going to join to form our citrate molecule, and then that's what's going to allow the citric acid cycle to run and generate our 3 NADHs that can then be used in oxidative phosphorylation, our FADH2 that can also get used in oxidative phosphorylation, and a GTP molecule as well. Right? So lots of energy that could be made from our acetyl-CoA. If we have lots of acetyl-CoA that's maybe coming in from our fats, our proteins, or our glucose. But I've told you that we have these other molecules called ketone bodies that can also get made from acetyl-CoA. So we have our carbs coming in. This is sort of our default pathway if we're thinking about what happens if you had breakfast this morning. So we've got our carbohydrates, our fats, and our amino acids can get broken down. So any excess are going to get broken down for energy to produce that acetyl-CoA. We just mentioned that we're going to be dependent on oxaloacetate. So that oxaloacetate is going to help us remove that the coenzyme A molecule in order to form citrate, and then that can enter into the citric acid cycle and generate energy in oxidative phosphorylation, plus that waste product, CO2, that we've talked a bit about in terms of that's where the carbons are actually going when you're producing these smaller molecules. If your mitochondria are functioning properly, remember that dependence on oxygen for generating all of that ATP at the end of the electron transport chain, that can then actually inhibit our pathways, right? So go back and look at the steps that are going to be regulated in the citric acid cycle. So high levels of energy can then slow down the citric acid cycle. And if we have lots of energy, what's going to happen to that citrate is we're going to export it and use it in fat synthesis. Okay, so if we then switch over to that anabolic pathway, we have lots of energy. We have a lot of these precursor molecules. We're going to store those carbons that either came from the breakdown of our glucose, any dietary fats, or extra amino acids in order to store them as our triacylglyceride molecules, as well as cholesterol that we mentioned. Ketogenesis is actually going to arise when we don't have a lot of oxaloacetate. Okay, and last day we talked a little bit about one of the sources of oxaloacetate, right? And those were carbohydrates. Okay, so your pyruvate can get converted to oxaloacetate. And if you're perhaps on a low-carb diet, then we might not have enough oxaloacetate to help supplement the citric acid cycle. Okay, it's still going to be running. You're never going to have no citric acid cycle running, but it does get limited in terms of how fast it can run. And so if you have a lot of fats and a lot of amino acids that are maybe getting broken down to acetyl-CoA, and we start to have concentrations that rise that the citric acid cycle can't accommodate in the liver or can't break down efficiently, that's when ketogenesis is going to arise. Okay, so we're going to see that those acetyl-CoA molecules instead are going to go and be converted to these ketone bodies. There's no energy required for the production of these ketone bodies. It's just going to be sort of another side reaction that'll spontaneously occur. So thermodynamically, you have lots of substrates that are going to drive these reactions in the forward direction and form these ketone bodies that will then get released by the liver. Okay, so these are going to get released into the bloodstream. And then other cells are going to be able to use them for ATP. Okay, so this is all going to be happening in the liver when we have a drop in oxaloacetate, which might be coming from carbohydrates. We had a huge influx of acetyl-CoA levels in our cells, in the liver cells, and that's what's going to drive the production of ketone bodies. All right, so if we look at ketone bodies, I've put ketone in sort of quotation marks. Can anybody figure out or identify why they're called ketone bodies, but these three molecules, that's maybe a slight misnomer in terms of calling them ketone bodies? Can anyone tell why? Yeah. The beta-hydroxybutyrate is not a ketone body. How come? Okay, so the answer is a ketone group is a C double bond O that's going to be attached to carbons on either side. So if we look at acetoacetate, we've got a ketone. Acetone as well has a ketone. Beta-hydroxybutyrate just has our carboxylic acid group on it, so it doesn't actually have its own ketone group. Remember, once you modify the functional group slightly, it has a new name, right? It's not a ketone anymore. It's now a carboxylic acid group. Okay, so out of the three, these are the three molecules that are going to get made. So remember, I said that acetyl-CoA, when our acetyl-CoA levels rise, we have a huge amount of acetyl-CoA. There's no ATP that's going to be required to make these molecules. Okay? The energy that we're actually going to use is in that thioester bond. Okay, so remember, we've talked a bit about the different types of bonds that are present. We've talked about phosphoryl transfer potential. That's where ATP has these phosphates, and you break and release that phosphate. That can help drive reactions. This thioester bond is also considered sort of a high energy bond, so when it gets broken and released, that's what's going to help drive the creation of these ketone bodies. Okay, and you can kind of see that structure. So here's our acetyl group, so your CH3, and your ketone, your C double bond O, and we can see those similar types of structures in acetoacetate, acetone, as well as slightly in beta-hydroxybutyrate. And we're not going to go through the reactions, but I will show them to you. But really just knowing that once you have lots of acetyl-CoA, we can use that thioester bond, release that coenzyme A molecule, and that's going to help drive or join these acetyl carbons together in order to form our three ketone bodies. So two of them can actually be used for energy. Okay, so they can get converted back to acetyl-CoA to generate energy. But we can see that they both have that carboxylic acid group in them, and when they're made, they also release a proton. So you might hear the term ketogenesis, so that's just making ketone bodies, which is normal for your body. It's normal for it to produce the ketone bodies when you have rises in acetyl-CoA. But you might also hear ketoacidosis, okay? And so that's the metabolic condition when you have way too many ketone bodies being made at any given time. And that's all because our carboxylic acid groups are going to release a proton when they're being made. How do we know that that proton is going to get released when either beta-hydroxybutyrate or acetoacetate is going to get made? Why didn't I draw that in the protonated state? How come it's in the deprotonated state? Yeah, in the back? I missed the last bit, sorry. Yeah, physiological pH, right? Let's go all the way back to the beginning of the course where we were talking about physiological pH, right? 7.4-ish. When you think of your pKa values of your carboxylic acid groups, remember they have low pKa values, so those functional groups are going to be deprotonated because your pH is way above your pKa value, and that's why they're going to get deprotonated, and when those protons get released and you have too many of them, that's what can actually contribute to acidosis. Our third ketone body, acetone, is a volatile compound. You might have used it or worked with it in maybe some of your labs or in chemistry, and it actually gets exhaled. So people might have some people say it's like a fruity breath, or it might contribute to maybe a little bit of that bad breath in the morning before you brush your teeth, all due to the breakdown of fats, generating a huge amount of acetyl-CoA in order to generate these ketone bodies in your liver. All right, so here's just another representation where you can kind of see the carbon skeletons as they're coming together, that release of our coenzyme A in order to first form acetoacetyl-CoA. Then a third ketone body comes in, so really we have, a third acetyl-CoA comes in order to form HMG-CoA, which we've seen before in cholesterol synthesis, so similar to what we saw in cholesterol synthesis. But again, you don't need to know these reactions, but I just wanted to show you how the first ketone body that's made is acetoacetate, and then there's also a redox reaction that will form that beta-hydroxybutyrate, or a decarboxylation reaction, which we've seen lots of times in biochemistry in order to form that acetone molecule. Okay. So really 3 acetyl-CoAs are going to come together. Those are going to generate our 3 different ketone bodies, so you might maybe make acetoacetate, and then that might get released from the liver, or the acetoacetate might spontaneously break down to acetone, or participate in another reaction to form beta-hydroxybutyrate. So you're going to have multiple levels of the various ketone bodies, and I think on that thinker slide I had a picture of ketone level tests that you can buy or purchase, and they're going to be actually looking for the levels of the various ketone bodies. So obviously not acetone because that gets exhaled, but the acetoacetate or the beta-hydroxybutyrate. All right, so our liver is going to spontaneously make these ketone bodies. They're going to get released into your bloodstream, and then your other cells are going to be able to pick them up and use them for energy. These molecules can cross the blood-brain barrier. So we've talked a lot about how fats can't get used by neuronal cells for energy, but the ketone bodies themselves are smaller. They don't have that CoA molecule on them, so they're able to be transported into neuronal cells and used for energy. Other cells like your muscle cells and your heart cells, those can also use the ketone bodies, so they can also bring them into the cell. And what we're going to see is that they actually get converted back to acetyl-CoA in order to generate energy. Okay, so we just see the reverse reactions, and again, you don't need to know the reactions that are taking place, right? But it is going to be sort of the reverse of its synthesis, where we're going to convert them back to acetoacetate, and then the addition of our CoAs is actually going to form 2 acetyl-CoAs. Okay, so this is kind of the important part, is that whenever acetoacetate or beta-hydroxybutyrate come into another cell, they can get broken down in order to produce 2 acetyl-CoAs. And we've already talked a bit about what molecule we're going to need. So if that acetyl-CoA is coming into, say, a neuronal cell, your brain wants to use the ketone bodies for energy, what molecule are we going to be dependent on? Oxaloacetate, good, right? So our acetyl-CoAs, those are going to enter into the citric acid cycle, right? Combining with that oxaloacetate. But where does this oxaloacetate come from? From glucose, and that's going to come from gluconeogenesis, right? So your liver cell is not only providing those ketone bodies, but it's also going to be providing some glucose to help your other cells process the ketone bodies or use the ketone bodies for energy. How much energy could we actually get out of 2 acetyl-CoAs? Can anybody do that? Yeah, off the top of your head? Not 14, higher than 14. Okay, how much? 20? It's a good guess because it's right? So each acetyl-CoA is going to make 10 ATPs. So if you have 2 of them, you're going to make 20 ATPs. So a little shorthand when you're doing your ATP calculations, if you want, right? For every acetyl-CoA that goes into the citric acid cycle, you're going to get 20 ATP or 10 ATPs. And if you have 2 of them, then you're going to get 20 there. Okay, so just make sure you go through and review your ATP calculations for the final exam. All right, so if we think about when this is all happening, so I said your liver may be more important than your brain. Your brain is ultimately the most important, right? It has the ability of shutting down the rest of your body to conserve energy for itself in extreme conditions, okay? But if we think about when this is actually going to occur, right, this is when we're going to be in the catabolic state, and in particular, maybe when we're under the glucagon hormone signaling pathway, so when we're in the low blood glucose state, right? We don't have a lot of carbohydrates, so we're not supplementing the citric acid cycle in your liver, and that's when these acetyl-CoA levels are going to rise. All right, so glucagon signaling is going to help generate glucose. It's also going to help with the catabolism of maybe your glycogen, catabolism of your fats, so your triacylglyceride molecules. Oops. Those are going to get broken down to produce our glycerol and our 3 fatty acids. And then that glycerol could maybe, as we learned last day, that could get used in gluconeogenesis. And then that's going to release glucose into the bloodstream. Okay, and so through glucagon signaling, I probably should have drawn that. We'll just draw a little receptor with it bound. That's our GPCR signaling pathway, right? That's going to help turn on gluconeogenesis in order to release our glucose. And then your liver might also be trying to supply maybe some of our glycogen being broken down in order to supply glucose as well. Right? So this is really going to deplete the amount of glucose that your liver has because your liver really is the provider for the rest of the body. It wants to share that glucose with the rest of the body. And one other way it can do it is it's also going to be using oxaloacetate. Okay, so remember oxaloacetate is one of the intermediates in gluconeogenesis, so it can also get used to produce glucose for the rest of the body. Okay? And when those fatty acids are going to get broken down to acetyl-CoA, if they can't combine with oxaloacetate because your oxaloacetate is being depleted, that's really when your ketone bodies are going to get made and released into the bloodstream. Right, and this is just really happening in your liver, but it's one way the liver can function in order to now supply maybe your brain with 2 really essential molecules that can then get used for energy. Okay, so I've drawn a brain, but you could consider this muscle cells as well. Your heart too are now going to use that glucose. Glucose is going to come in. We're going to break it down to pyruvate. That pyruvate can get converted back to oxaloacetate, and then your ketone bodies will get converted back to acetyl-CoA. And then those are going to combine in the citric acid cycle to eventually make ATP. Okay, so really working together because your brain can't use those fats, so the fats can't get in. Just put a big red X. Really dependent on glucose for energy, as well as helping supplement the citric acid cycle, and then those ketone bodies can now come in and also get used for energy production. Questions about this? Or do you kind of see how everything's coming together? It's getting integrated. What's unclear? Yeah. Beta oxidation. So where's the beta oxidation happening? Where it can't happen in the brain, but it's going to happen in your liver cells. So here's your beta oxidation, right? This fatty acids to acetyl-CoA, that's where beta ox is going to take place. So that's still taking place in the liver cells, generating those acetyl-CoAs, it's also going to generate some ATP, right? Your remember, gluconeogenesis is dependent on energy, or ATP, so beta oxidation is still going to help generate some NADH, some FADH2s. Those could get processed by the electron transport chain and oxidative phosphorylation to help supply that energy that gluconeogenesis is now going to use. I think I saw another hand or question, yeah? Yeah, so the key point is the oxaloacetate is not going to be able to be used to process the acetyl-CoA because it's getting pulled out in gluconeogenesis. Well, they are happening at the same time, or you could maybe think of gluconeogenesis happening first, depleting and pulling away all that oxaloacetate, and then that's why ketogenesis is going to arise. Okay, so gluconeogenesis uses up the oxaloacetate, citric acid cycle can't run, now your acetyl-CoA levels rise, and that's when ketogenesis will occur. Yep. Glucagon, low glucose state, we also can't make oxaloacetate from glucose. Glycolysis will definitely be turned off. Yes, when gluconeogenesis is on, glycolysis is going to get turned off by that tandem enzyme, as well as phosphorylation by the GPCR signaling pathway. All right, so really this is going to happen when we've got lots of fats being broken down. Maybe we have some of our ketogenic amino acids are going to be broken down as well. Our processing of the acetyl-CoA is going to be limited in the citric acid cycle due to low levels of oxaloacetate. That can happen when your glucose levels drop, and instead our ketone bodies are going to get synthesized for energy production in other cells. That's kind of the key point, right? Our two ketone bodies that can be used for energy are acetoacetate and beta-hydroxybutyrate. They'll both get converted back to acetyl-CoA in either your brain, heart, muscle, or even kidney cells. And that third ketone body, acetone, is going to be a waste product that's going to get exhaled. Okay. If you have too many of these ketone bodies being made, because they are acidic species, your acetoacetate and your beta-hydroxybutyrate, this can actually lead to a drop in blood pH as well as acidosis. So coming back to our buffers that we talked a little bit about at the beginning of our course, when we have lots of ketone bodies being made, that's where ketoacidosis can occur, right? Either under low-carb diets, maybe under starvation too, where you start to break down fats, and we'll talk a little bit about how that fits into diabetes as well. We usually have this blood buffering system, so bicarbonate, that can help with absorbing all of those protons, forming carbonic acid, and then breaking down to a water molecule and a CO2 molecule. And one of the symptoms you might notice in somebody that has acidosis is this weird exhaling breath. Okay, so their body is trying to get rid of all of these protons, where CO2 eventually is going to get made, and so they might have this weird sighing breath that might be one symptom or one indication that they are in the acidotic state. In physiology, you might have also learned about how bones and different tissues can help, as well as the renal secretion. But really, what we want to focus on in this course is that acidosis in terms of what effect it can have on other molecules in the body. Okay, so if we don't have a lot of bicarbonate to help with this buffering, right, your blood pH might drop, maybe below 7.35. And my question for you is, why is this bad? Okay, so let me maybe take a minute or two, discuss it with a partner, and I'll just throw up the Mentimeter question once the computer wakes up for me. All right, so we've got some things coming in, right? Coma or death, you might end up dying, unfortunately. That can happen, right? And some sort of better answers. So we're going to disrupt the metabolic processes, but how, right? How are we going to disrupt the metabolic processes? We have lots of proteins that are enzymes, right? And they are dependent on their overall structure. So they might be denatured. That might lead to lower enzyme efficiency, right? Your enzymes might have optimum pH values, right? So their active sites might have optimum pHs. So they're not going to be functioning. They won't be able to catalyze those reactions if there's a drop in pH, right? We could see a decrease in oxygen carrying capacity, yeah, so that's definitely the function of the protein, right? So if you change the structure, you're going to change the function, okay? That structure-function relationship is really important, and hopefully one of the main things that you take away from this course in terms of proteins. The transport of molecules might be inhibited across the lipid bilayer, maybe, yeah, if you're talking about a membrane protein receptor that's important for transport, right? Reactions might be lowering the pH in the lysosome. Maybe the lysosome does have a low pH, and that could affect it, but really it's the destroying of proteins and affecting their function that's going to influence our metabolic pathways. Okay? The other thing I guess that we didn't really talk a bit about is maybe binding, right? So all of those non-covalent interactions, so maybe your H bonds, maybe your ionic interactions, right? All of those interactions are important for the protein's structure, so you might have the whole protein denaturing, or you might just be protonating different regions of the protein that might be important for binding, and so then you might not be able to transport different things, either in and out of the cell, through the bloodstream, or even binding interactions, all those protein-protein interactions that Dr. DeMello went through earlier on in the course. All right, so your liver really is going to be important for supplying different molecules, both glucose, either from glycogen breakdown or gluconeogenesis. We've got different molecules that can be synthesized in the fat in the liver, like your fat synthesis and your cholesterol synthesis. Last week we talked about lipoproteins that are going to get released into the bloodstream. But when it comes to energy production, your brain can't use those fatty acids, so that's where these ketone bodies from acetyl-CoA is going to be really important. So either arising from that fat breakdown that's happening in the liver, maybe some ketogenic amino acids, so if you're in sort of a starvation state and proteins are starting to be broken down, those are going to help supply amino acids both for ketogenesis and gluconeogenesis as well. Okay? So definitely a really important organ that you should definitely be taking care of, very helpful in terms of supplying these fuel molecules for the rest of your body. All right, and so if we think about when ketoacidosis can arise, remember we are in the catabolic state, right? So for patients that might have type 1 diabetes that are unable to synthesize insulin and have to take synthetic insulin, if they're not taking any insulin, their body's going to switch over and be in the catabolic state and try to start breaking down those free fatty acids. Okay, so if we don't have insulin coming in, then our glucose levels are going to drop. Our fats are going to be released. We get that fatty acid mobilization, remember, transported along albumin. They might come into the liver in beta oxidation, producing that acetyl-CoA. But if we don't have the oxaloacetate due to an absence of glucose, that's where ketoacidosis can arise and ultimately could lead to coma or death in somebody with type 1 diabetes. So definitely something to think about, right? If you don't have those glucose transporters, you know, this could be something that arises in somebody that has type 1 diabetes, right? They're not able to import that glucose, and instead they're going to switch over to fat catabolism in order to try and make some energy from all that acetyl-CoA. But in the absence of pyruvate and oxaloacetate, the citric acid cycle is going to slow down, and instead those ketone bodies are going to spontaneously form in the liver. If we have too many of them that are being made and that bicarbonate levels drop, then we're not going to be able to buffer them, and we get a drop in blood pH, leading to ketoacidosis, the production of extra CO2 from that bicarbonate buffering, and then eventually affecting protein structure and function, maybe some of our enzymes, and that's where that coma or death can arise, right? If your metabolic pathways are affected, they're not able to make energy for the cells, then that's when you're going to see that cellular death, and ultimately unconscious, maybe trying to conserve some of that energy for the brain, okay? There are a couple symptoms, and what I want you to start thinking about is how can we tie back symptoms that we might be seeing to the metabolites, to the biochemical pathways that are taking place, okay? So I'm going to give you 2 minutes to talk with your partner and thinking about what's going on in the body. If someone has type 1 diabetes, what's going to be the underlying biochemistry between these the 4 T's? So these are the 4 things that you might notice in somebody with type 1 diabetes. So start keeping an eye out on it in case somebody might have undiagnosed diabetes. These are things that you might notice. They might be going to the washroom quite often, they might be tired, they might be very thirsty, and they might also be getting thinner. Okay, so if you've talked about this in your physiology classes, try to tie it back to the biochemistry and see if you can come up with the link there. All right, we've got some answers coming in on Mentimeter in terms of the various symptoms that we're seeing. Okay, so in terms of that toilet, right, we've got excessive urine, so your kidneys are going to be trying to excrete maybe glucose. So if they're hyperglycemic, your body is going to be trying to get rid of it through the urine. We're also going to see ketone bodies, and maybe even those acidic species that are going to help try to get rid of it. So that's going to really affect your kidney function, right? And sort of related to that is that thirst. So they might be dehydrated, right? If you have lots of osmolarity, so we've got lots of molecules in the bloodstream, right? So you're going to have increased blood osmolarity. That's going to pull the water out of your cells, leading to dehydration. So that might lead to that thirsty symptom. Down here we've got thinner because the body's less able to absorb nutrients. So I guess we're talking about that glucose, right? But we're actually going to be breaking things down, right? So we're not going to be able to make enough ATP. So there's the decreased ATP from either glucose, maybe from our fats too, and the thinner is really going to come from that fat breakdown and beta oxidation. Okay, so start to be able to tie back things that you're seeing in terms of symptoms, in terms of conditions. Try to relate it back to metabolic pathways or defects that you might be seeing at the cellular level with various conditions. Going forward, because that could definitely be helpful to recognize if some of these symptoms may have some other underlying cause in the body. So when we're talking about someone with type 1 diabetes, right, we talked about having high blood glucose, kidneys trying to excrete it, but as soon as they take that insulin, the synthetic insulin, that can really help reduce that hyperglycemia. Glucose transporters get recruited, and then the glucose can get broken down to make ATP. Things like polydipsia, so that extreme thirst or polyuria, going to the toilet, are going to be eliminated due to that drop in blood tonicity, so the glucose gets used up. And then ketone body formation might also go down, right? You have glucose coming into the cells. That's going to help make that oxaloacetate to process all of that acetyl-CoA. Our blood tonicity normalizes and your water balance as well. Okay. And now that we can use that glucose, we're going to be using it in glycolysis, so glycolysis is going to get stimulated. And then gluconeogenesis and glycogenolysis will be inhibited. So remember your competing pathways as well there. Our fat breakdown is also going to get inhibited, while we can maybe switch over to our anabolic pathways with fat and glycogen synthesis rising from those excess carbon molecules. I just wanted to throw this up just to show, I mentioned it on Tuesday in terms of that amino acid processing. There are 2 blood tests that might be done that you might also get measured, so AST and ALT. These are enzymes that are normally in your liver, so if they are not in the liver and they're in your blood, then that tells you there might be something wrong with your liver. So there's different types of liver damage where these cells can get damaged and their cellular contents get released. So if you're ever going in for a blood test and they want to look at those 2 levels, then that can give you an idea of different enzymes that may or may not be functioning properly in your liver. Okay, so there's lots of different metabolic pathways that are going to be really important throughout your body, the flux or the movement between catabolic pathways or anabolic pathways, they're all going to be regulated by thermodynamics. We've seen lots of reactions that are essentially irreversible.